You know, go home for 100 and bring opening. Eight years. Who was that?
Okay, so today we're going to be talking about the somatic nervous system and starting to think about how you go towards actually voluntarily moving your body. So before we start, I want you to jot down a quick couple definitions for yourself. So these are some words we've seen before. So make sure that we are prompting ourselves, prepping our minds for thinking about the words afferent, efferent, remembering what an EPSP is, and what depolarization is. Like 2 minutes to do this. If you're fast and get bored, feel free to draw them out. So we'll come back to those in a second. But I did have a couple endocrine questions for you last time that didn't work. And in case you're waiting on those, I'll make sure we get a chance. It'll be a good review of Friday for us anyway. So go ahead and join us on our short quiz on some of these endocrine concepts we covered last time. So I think we got insulin and glucagon basically. All along. Okay, so to start with, oops, that's covering our question. All right, so which hormone is responsible for regulating blood glucose during the absorptive state? Yeah, so we got insulin as our hormone of the absorptive state. All right. Now, where's that insulin coming from? All right, so sweet. Our pancreas has different types of cells. Exocrine part is for enzymes. Alpha and beta cells are doing insulin and glucagon. The beta cells specifically are where our insulin is coming from. So insulin and beta cells in the pancreas during the absorptive state. All right, let's tack on some function. What's that insulin doing? So insulin does a lot of stuff. Which of these is not a thing we should associate with insulin? Okay. All right. So the correct answer is that insulin is not causing glycogenolysis, but it is doing everything else, right? So absorptive state is all about building stuff up, storing things in large macromolecules because we have an excess of energy and excess of glucose coming in. So we're pulling glucose in the cell, right? The first one, we are making fast-making triglycerides out of extra nutrients coming in. We are also making proteins out of extra nutrients coming in, but what we're not doing is breakdown. So we're not doing this glycogenolysis because that's breakdown of glycogen that would release glucose. We don't need to do that because we've got plenty coming in during the absorptive state. So finally, what was our trigger for this insulin release? Okay, yeah, our trigger is when we know we have just eaten something and it's in our guts, we're absorbing it. So specifically, what we're going to see is an increase in blood glucose at the beginning as we're getting ready to start our absorptive state, as we're getting ready to release insulin. You ate something, we've got glucose in the bloodstream. Now we need our signal to the cells to absorb that glucose and to start building things. So that's our story of insulin, right? We enter the absorptive state because we're absorbing things, particularly glucose, storing them for later. Okay. Great. We would walk through the opposite if we were thinking about glucagon. Okay. So we want to be able to tell where our hormones come from, why we're releasing them, and what they do. So back to our somatics, right? So that's the end of our endocrine system, end of understanding our hormones. And now we're moving on to thinking about muscle. So we're thinking about a different type of effector organ now. Endocrine glands, they're part of our list of different effector organs for our autonomic nervous system. And we have cardiac muscle, smooth muscle, and glands, right? So all of those things would be sympathetic, parasympathetic innervation. Now we're moving towards our skeletal muscle, different type of effector organ, specifically for the somatic nervous system. So when we're orienting ourselves to the somatic nervous system, we want to know where we fall kind of in our nervous system map. So we want to remember our afferent and efferent nerves because those are telling us kind of directions of information. Our afferent nerves were ones that are out in the peripheral nervous system, but their job is to send information into the central nervous system. So our afferent nerves are bringing information from receptors, sensing touch, sensing stress, sensing pressure, sensing chemicals out in the body. Tell your brain what's going on. Right? That's our afferent nerves. And our efferent nerves are those nerves that go out and do stuff, right? So coming out of the central nervous system, going out into the periphery, telling us, okay, start releasing these hormones or start contracting a muscle, depending on what type of nerve we're on. The reason we got our EPSP and our depolarization on here, because as we think about the somatic nervous system, we're going to be thinking about linking up a neuron, right, to its junction, to the point where it meets a muscle cell, right? So we want to remember our EPSPs are excitatory postsynaptic potential, right? So we know that we can depolarize, so make more positive that membrane potential in our next cell in line in a pathway, right? Before we were thinking about neurons, we're actually going to switch and think about muscles forward. So we can make it more likely to set off another action potential in our next cell if we've got one of these excitatory postsynaptic potentials. So we link that in with depolarization because depolarization going up, right, in terms of our charge is how we get towards that action potential. So same sort of situation with C and D, different emphasis on what we're focusing on. So here's that nervous system map. So remember afferent nerves coming in, right? So that sensory information, our efferent nerves going out. We talked about the autonomic nervous system, our rest and digest part, that parasympathetic division, our fight or flight part, sympathetics, all going to our smooth muscle, cardiac muscle, and gland in order to maintain homeostasis in the body. So now we're peeling away and we're thinking about the opposite of the autonomic nervous system. We're still sending signals out. These are still efferent nerves. But now we're focusing on how do we get to a skeletal muscle? How do we get to one of these voluntary muscles, one of these ones you can actually think about and contract? What are the nerves that get us there? What do they look like? So they're going to look a little different than when we look at our autonomic nerves. So we're going to be thinking about that somatic nervous system, and we're going to be focusing in on the point where those somatic nerves join up with our muscles. So that's going to be our neuromuscular junction. So it's going to be similar to a synapse, but now we're kind of like changing the tissue types. So we're going to change some terminology. And then we are going to move into thinking about, okay, what happens when this goes wrong? We have some toxins that can affect the neuromuscular junction and can help us think about what the different parts of the system are doing. And then if we have time, we'll move into thinking about the structure of a skeletal muscle and how we start to contract it. Okay. So let's take a look at one of those somatic nerves. So our somatic nerves are efferent, going out from the central nervous system. We also often refer to these ones as motor neurons. We could call any efferent nerves motor neurons, but because we associate motor with motion and we think about the voluntary motion, right, it makes most sense and it's used most commonly when we're thinking about somatic nerves. Yeah. So our somatic nerves are going to be starting in our spinal cord. Right? So here we see a cross-section of the spinal cord. Here we are in the ventral horn. So we're on the belly side in this projection here. Okay? We can see this motor neuron, this somatic nerve starting and heading out, ultimately joining a spinal nerve. So coming out some level, vertebral level, so some level of your spinal cord, and then heading to a skeletal muscle. Okay? So when we get to a skeletal muscle and connect to our muscle fiber at our eventual neuromuscular junction, we're going to see that we're going to have receptors in the skeletal muscle, and we're going to have neurotransmitters coming out of that somatic nerve. So like we saw in lots of places in our autonomic nervous system, acetylcholine is above here, right? So we saw acetylcholine in our parasympathetic nervous system when we're talking from preganglionic to postganglionic fibers. And then even when we were talking to the effector organs for the parasympathetic nervous system, we saw acetylcholine even when we were thinking about sympathetic nerves, we were between preganglionic and postganglionic fiber. That was still acetylcholine doing its job. Okay. So now in our somatic nerves, again, we see acetylcholine. Now the type of receptor that we're going to see is the nicotinic cholinergic receptor. So that's what's going to be on the somatic nerve. That's that fast type. So we see the nicotinic cholinergic receptors also between preganglionic and postganglionic fibers in our autonomic nervous system. Our other types were the muscarinic cholinergic receptors. Those were only at the end of our parasympathetic nervous pathway on those effector organs. So acetylcholine is the most common neurotransmitter we're talking about in this unit, and nicotinic cholinergic receptors are the most common of our two types of cholinergic receptor. Hopefully these are familiar, friends. Okay. So when we look at the structure of a muscle and how it links in to the neurons coming through the somatic nervous system, we're going to see this concept of a motor unit. Okay. So if we trace any given motor neuron, so we're going to trace motor neuron 1 up top here. So if we trace this motor neuron, we're going to see it heads out into a spinal nerve, comes down towards a muscle, and then we see it splitting, right? So we see it splitting, and we see it connecting to a muscle fiber here. Muscle fiber A, let's say. We see it connecting to another muscle fiber over here. That would be, right? And we got some more muscle fibers over here, right? So we might call this one C, and this one looks like it's wrapping around the side to get us to D over here. All right, so we can see that one motor neuron went to multiple muscle fibers. Okay? So when that motor neuron 1 sends off an action potential, the action potential heads down the axon, it's going to split, and it's going to go out to all those endpoints. So it's going to get to all those muscle fibers from just that one motor neuron. So all those muscle fibers are going to contract at the same time. They're going to contract together. So we call them a unit, right? So that is our motor unit there. So it's one motor neuron plus all of the muscle fibers that it's innervating, right? Now, we have multiple motor units in any given named muscle, right? So if we're thinking about your biceps brachii, right, you'd have multiple motor units. You'd have multiple motor neurons going there. So you'd have different parts of the muscle contracting together, so different fibers within the muscle. So we're always going to skeletal muscle if we're on a somatic nerve. We're always going to multiple muscle fibers, right? Any given muscle fiber, though, is only going to have one neuron you trace it back to, right? So unlike when we were talking about convergence of different neurons on postsynaptic neurons where you might be getting signals from multiple places, we don't have to worry about that for muscle fibers. Muscle fibers, any given muscle fiber, just traces back to one of our neurons, right? So we don't have conflicting signals when we're thinking about a muscle. All right. So if we zoom in on that connection between our motor neuron from the somatic nervous system and the muscle fiber that it's innervating, what we're looking at is our neuromuscular junction, right? So neuro for the neuron, muscular for the muscle, and junction because they are connecting up. So at the neuromuscular junction, right, we're going to be thinking about our signals coming in. All right, so we are going to be thinking about our excitatory postsynaptic potentials or inhibitory postsynaptic potentials. So we're going to be thinking about depolarization in particular, getting us towards sending off a new action potential. Okay. And we're going to be thinking about that as a response to acetylcholine ACH, that hormone here. Okay, great. So we are going to be thinking about this now, though, in different terminology. So instead of thinking about it as a postsynaptic potential, we're now thinking about our end plate potential at a motor end plate. So similar concept, but different type of cell, so some different words. Good. So looking at the muscle side on that neuromuscular junction, and essentially we have a synapse between our motor neuron and a muscle fiber. Okay? We're changing names, right? So our axon terminal from the neuron coming in, we now would refer to as the terminal bouton. So we've still got the word terminal, right? So that's going to tell you that this is that same thing, just now specifically a neuromuscular junction. Okay? And the motor end plate is going to be the specific part on the muscle where we have this junction occurring. Right? So think about like a specialized type of membrane where we're going to have like receptors for acetylcholine here. So all of our motor neurons, all of our somatic nerves are releasing acetylcholine. And on that muscle side, we've got nicotinic cholinergic receptors receiving that acetylcholine signal. Right? So these are working in this ionotropic receptor way. So like our ligand-gated ion channels, right? So here we have our neurotransmitter coming in. So this would be acetylcholine. We're thinking about our neuromuscular junction. Right? And this is going to open up a channel, right? And that channel is going to let ions move through. A relatively simple pathway, relatively quick reaction here. It's good, right? You want to have like lightning fast reflexes in certain situations, right? You don't want to have to wait around for your skeletal muscle to respond to stuff. Okay. So these are going to be excitatory reactions when we open these channels. We respond to acetylcholine. We are going to let positive ions move in. Right, so our end plate potential on the muscle side at that motor end plate, right, is going to be depolarizing. Right, we're going to see excitation happening as we connect from that somatic nerve to our muscle cell. Ultimately, we're going to see a different enzyme breaking down, or a specific enzyme, I should say, breaking down acetylcholine. So that would be acetylcholinesterase. Okay. Let's zoom in specifically on what this communication looks like now that we've kind of related it to some things we've seen before. Okay. So what we got here? Series of steps we're still going to need, like in all our other nerves, to trigger an action potential in that somatic nerve, right? So we're receiving a signal from somewhere in our central nervous system. Most frequently, we're coming from the spinal cord, right? And we're heading out on this motor neuron, sending an action potential. So this wave of depolarization coming down that motor neuron. So now here we have that use of our new terminology. So instead of calling this our axon terminal, we're calling this the terminal bouton. So it's like a button, right? We've got this little button coming off the end of our motor neuron, linking us up to a muscle fiber. Okay. So in that terminal bouton of the motor neuron, we have a reaction that looks like what happens in our synapses, right? So just like in our other types of synapses, we would see, zooming in a little more, voltage-gated calcium channels. All right, so when we have that depolarization to our terminal bouton, right, that's going to trigger our release, or sorry, our opening of these voltage-gated calcium channels. Calcium always wants to flow in, so it does when we open these voltage-gated calcium channels. Just like in the synapses that we talked about before, that's going to trigger ultimately the release of this neurotransmitter, which we now know is specifically acetylcholine. Okay? So that calcium is entering the cell. We had our acetylcholine stored in these little synaptic vesicles. So we've got little baggies of acetylcholine waiting for the signal. Calcium comes in, and we have that acetylcholine baggie merge with our plasma membrane and get released into this space in the neuromuscular junction. Okay. So now we've got our neurotransmitter, our acetylcholine, here in this space between our neuron and our muscle cell. So as it's floating around, it's going to bump into some of those nicotinic cholinergic receptors. So we can see that here, that binding of our acetylcholine to this nicotinic cholinergic receptor, which is wide open. Okay, so that's our key for this channel. And here we can see that that nicotinic cholinergic receptor allows both potassium and sodium to flow. We know that there's a really big gradient, a really big ECDF for our sodium in particular to come into cells. Our electrochemical driving force on sodium is really strong. So on net, what we have is lots of positive charges coming into the cell in the form of sodium. So we've got this net movement of sodium in. So this is a depolarization, right? So we're down somewhere negative when we start out. We don't have to think about specific numbers when we're thinking about our muscle cells, right? But we're going to come up. We're going to come up more towards zero. We're going to be depolarizing. And so that's our end plate potential, our EPP. Okay, so these positive charges coming in are EPP. Okay. This spreads around our muscle fibers. So you can see this forming a current. It's just the movement of charges, right? That's all a current really is. Movement of charges side to side, right? This is going on, depolarizing the rest of this membrane, right? Ultimately forming an action potential in the muscle cell itself. So muscle cells are another type of excitable cell, right? We talked about how neurons, when we say they're excitable, right, what we really mean is they can form action potentials and have a response to action potential. Now we're seeing that our muscle cells are also excitable. So we can have action potential in muscles, just like we can in neurons. They work a little differently in a muscle fiber, but we still have reactions to these changes in charge. So this action potential is going to travel across our plasma membrane here, and we are going to come back to it later when we think about what exactly the muscle fiber does in response. Spoiler here, eventually our muscle fiber is going to contract, right? So that's really the goal with this action potential, is to tell this muscle fiber to start contracting. But there are a lot of in-between steps to get that contraction happening that we'll focus in on as we focus in on the structure of skeletal muscle itself. Okay. So our action potential is going to head off. Eventually our muscle fiber is going to retract and contract in response. We also want to wash away the signal. Okay. So to wash away the signal so that you're not stuck in a contracted state forever, that wouldn't be good, right? Like if your muscles contract and never relax because that acetylcholine is constantly there, you're basically paralyzed, right? You got things like lockjaw situation, and you can't open your mouth if you can't relax those muscles. Okay, so we need to remove our acetylcholine. So here's our acetylcholinesterase, that enzyme on the muscle side. So before we just said in synapses that there are enzymes that break down neurotransmitters. Now we're naming one specifically. So here's our acetylcholinesterase that breaks down our acetylcholine, breaks off that acetyl group, leaves us with choline. We then have a reuptake molecule that pulls that choline back into your neuron, back into the somatic nerve, so you can make more acetylcholine. So you can just do this again, be ready for later. So different words than the steps of our synapse, but same idea. We're sending an action potential in order to release a neurotransmitter. When we release the neurotransmitter, we've got a receptor receiving that signal, and we have a response in the cell. Now we know that in muscles, it's going to be pretty simple. This is going to be excitatory, get us towards contraction. And we've got new vocab terms to learn. I recommend putting those on flashcards, but conceptually, this works pretty much the same. Let's take a peek at what this looks like. All right. Oh, look at the pictures. All right, so we had our action potential come down to the motor neuron. Now we're looking at the neuromuscular junction. That's a synapse on this motor end plate. Looks a little different than synapses did before. Now we got our steps. There's our action potential. That's our calcium coming in. There we see that acetylcholine being released. Now we're looking at the motor end plate. There we've got our receptors. They open. So just ions moving, no actual neurotransmitters crossing. You can see all that sodium coming in, making this potential less negative. There we're going to have our action potential in the muscle fiber. This removal occurs in two ways. One, acetylcholine diffuses away from the cement. There we have some acetylcholine floating out. There we have breakdown of acetylcholine into its component parts. And we pull the choline back in so we can make more. All right. Really? Okay. So when we think about our different parts of our nervous system, when we think about these different branches in the peripheral nervous system, we're going to want to be able to tell apart our two divisions in the autonomic part of the nervous system versus our somatic nervous system. So we're going to want to be able to tell where these nerves come from. We're going to want to know what the pathways look like. So didn't point it out, but you might or might not have noticed that when we were thinking about our autonomic nerve, we always had preganglionic, postganglionic, then effector organs. When we were talking about our somatic nerves just now, I think we have one, just one neuron going directly to our skeletal muscle. Right, so we have different effector organs for our autonomics versus our somatic part of the nervous system. We've got different receptor types, right? So we've got cholinergic receptors in our parasympathetics At the effector organ, those are muscarinic cholinergic receptors. So they're G protein coupled. We can see slower reactions and different cell responses in our autonomics. Our sympathetic autonomics, we had our adrenergic receptors responding to epinephrine and norepinephrine, a bit of complexity there. And then in our somatic nerves, we've got our easy type of cholinergic receptor, the nicotinic one. Right. Overall, we have different effects on our effector organs in our parasympathetics and sympathetics. We could either excite or inhibit our next neuron in line, that postganglionic neuron, or our effector organ, right? And in our somatic nervous system, when we get to that muscle, we're just going to excite it. Our autonomics were involuntary. Our somatic nerves, and therefore our somatic muscle, are going to be under voluntary control. So we want to be able to differentiate all these things. All right. So quick question to make sure we are awake. Right, so what neurotransmitter was it that we're releasing here from our motor neurons at our neuromuscular junction? What type of receptor do we then have on our motor end plate? Okay, great. Yeah, so acetylcholine coming out of our motor neuron, binding into our nicotinic cholinergic receptor. Actually, never going to see acetylcholine bind into any type of adrenergic receptor. Adrenergic receptors always have to respond to either epinephrine or norepinephrine. Right? Their name is coming from the adrenal gland, so like adrenaline is epinephrine. Okay. So same thing, epinephrine would never bind to a cholinergic receptor, because cholinergic receptors are specifically for acetylcholine. That's in the name as well. Right. And you might see epinephrine bind to an alpha adrenergic receptor, potentially, but that would be our sympathetic autonomic nerves, not our somatic nerves. Okay. If we think about the neuromuscular junction and how it works, one of the things that can help us focus in on different parts of the system is how things go wrong. So we're going to take a second to look at some natural toxins that create problems with your muscles because they affect how we release acetylcholine and what happens when we do. I'm going to ask you first, what do you think would happen to a muscle if from our somatic motor neuron, we end up releasing extra acetylcholine? So a lot more acetylcholine than we normally would. Okay, great, 100%. Yeah. So if you have these extra acetylcholine, if this is going to be a really strong depolarization signal, a lot of end plate potential here, lots of action potentials in that motor muscle fiber that we're attaching to. So this can cause muscle spasms if we release too much acetylcholine. Okay. So we have neurotoxins that do this. So what we're looking at here, all right, we're going to think about three different types of toxins. So if we think about latroxin here, right, this is what happens, right? So when we get bit by a spider, the reason that we have spasming and paralysis happening is we have an extra stimulation of that acetylcholine. Right? So extra acetylcholine means extra opening of those nicotinic cholinergic receptors, and therefore extra current, extra action potential, extra contraction happening in the muscle responding. So this can cause muscle spasm, right? And ultimately, it's going to cause your body to go rigid, right? As these muscle spasms and muscle contraction continue, right? And maybe that's just annoying if it was in your leg or something, but if you think about what that would do if it affects your breathing, right? This can kill you, right? Now, in the case of crotoxin, we've got a different problem, right? So in this case, we inhibit acetylcholinase. So I don't have a question for you, but I want you to take a second in your mind and think about what you predict is going to happen if we inhibit our release of acetylcholine. So let's trace through the steps in our mind. Yeah, so if we get bit by this snake over here, right, and crotoxin is released, we inhibit that acetylcholine, right? So say we don't have acetylcholine in this synaptic cleft here, in this neuromuscular junction, when we should be, right? So you're sending out your action potentials down your somatic nerve, down your motor neuron, right? You're trying to contract your muscle, but you can't release this neurotransmitter. This is a new way of being paralyzed, right? So instead of being rigid, these muscles have what we refer to as flaccid paralysis. So they're relaxed. You can't contract them because you can't you can't secrete this neurotransmitter here. So this is likewise a problem. Contraction is also important to things like breathing. We have skeletal muscle responsible for that. Okay. Finally, what do you think is going to happen if instead of affecting the release of our acetylcholine, we act on the receptor side? So let's imagine we have our nicotinic cholinergic receptors and they get blocked. Okay, great. Yeah, so same situation as if we were inhibiting that acetylcholine release, right? So in this case, from our neurotoxin curare, right? What this does is it blocks cholinergic receptors. And so we see that flaccid paralysis, that extra relaxation again. But we're affecting a different part of the junction, even though we have the same effect. So we're going to brief intro now the actual structure of our skeletal muscles so that next time when we meet, we can focus on the process of contraction, which is a little chunky. Okay. When we're thinking about a skeletal muscle, you've seen them in lab, but you might not have noticed all these different layers because some of them are microscopic. Okay. So surrounding any named skeletal muscle, right, you're going to have this layer of tissue called the epimysia. Okay, so kind of clear. You may or may not have noticed that on some of the muscles, there's a little extra kind of like clear, shiny coating on them versus the ones that look potentially a little rougher, potentially darker colored where we peeled it away. Okay, so we got some connective tissue around it. Okay. We also have connective tissue that extends into the body of the muscle. Okay. So if we extend into the body of the muscle, right, this is our perimysium. Okay. And that actually surrounds little groups of muscle fibers, right? So we can circle these out in this diagram, right? And each of these little bundles we'd refer to as a muscle fascicle, right? So fascicle isn't still at the level of a muscle fiber, right? So this isn't a motor unit. This isn't an individual muscle fiber. This is still a larger scale. You're going to have hundreds to thousands of muscle fibers within any given little bundle of, within a little bit bundle, the bundle is a fascicle. Okay. So we got to zoom in even further to get where we're going as we think about how do we contract a muscle fiber. So as we go further in, we are then going to see our muscle fibers within that fascicle are surrounded by endomysium. So this is still connective tissue, but we're even deeper in. So endo, think like inside. So we're even more inside the muscle. And then it's not until we're within that endomysium that we're going to zoom in on individual muscle fibers. So here we're pulling out a muscle fiber, an individual cell of our skeletal muscle. So you're going to notice looks different than a normal cell, right? Like our muscle fibers are really long. Not true when we look at a lot of other types of cells. And we're going to notice that we have lots of nuclei here in this muscle fiber. So one of the big differences between muscle fibers and a lot of other cell types is we don't just have one nucleus, we have many because of how these muscle fibers form. Because we're going to have specialized function in the muscle fibers and how their membranes function, we give their plasma membrane a new name. Okay, so when we talk later about the sarcolemma, that's our new name for the plasma membrane on our muscle fiber. Okay, so still a membrane, but we're going to see looks a little different, functions a little different. Okay. And because our inside of the muscle fiber also works in a very specific kind of different way, it gets a new name too. So our sarcoplasm is basically our cytoplasm for our muscle cell. So anytime you see sarco, right, or anytime you see this mycium, my sort of root in a word, be triggering, this is probably something to do with the muscle. So next time we'll pick up here, and things to look ahead at, if you want to be prepared, I would recommend we're going to be looking at the structure of sarcomeres, and hopefully we will get into understanding our cross-bridge cycle. So if you're going to take a look at 2 figures before class on Wednesday, I recommend those 2.